Khateeb series in Human Anatomy and Physiology. Chapter 2, Cell Structure and Function. Episode 1, Introduction to Cell and Plasma Membrane. Welcome to this engaging session on cell structure, transport, and division. Today, we will dive into the fundamental aspects of how cells operate and interact with their environment, uncovering the intricate details that make life possible. Together, we'll explore the structure of the plasma membrane and how it defines the boundaries of the cell and regulates what enters and exits. We'll then unravel the essential functions of the principal organelles within the cell, each performing a vital role that contributes to its overall operation. Next, we'll delve into the fascinating processes of mitosis and meiosis, discovering how cells grow, repair, and reproduce. Finally, we'll compare and contrast the different types of cell transport, enhancing our understanding of how substances move across cell membranes to keep the cell nourished and functioning optimally. Cells are the fundamental building blocks of all living organisms, performing essential life processes through their dynamic structure and specialized functions. These cells are tiny structures composed of specialized organelles, each to carry out a specific task such as DNA replication, energy production, protein synthesis, and waste disposal. Imagine each cell as a tiny, self-sufficient factory, where every organelle functioning like a machine within this factory to perform special tasks to produce the products. But the important question is that how do these remarkable units come into being? Picture atoms, the basic elements like carbon, hydrogen, oxygen, nitrogen, and phosphorus joining together to form molecules. These include as water, proteins, fats, sugars, and DNA. These molecules then organize into organelles, which in turn unite to create the cell, a dynamic hub where life's essential processes occur in harmony. At the very start of human life, this marvel of organization is embodied in the zygote, a single cell formed when the sperm meets the egg, merging genetic material to create something entirely unique. Almost immediately, the zygote begins to divide through continuous stages. This gives rise to an embryo, which is a collection of cells that eventually grows into a fetus. This incredible progression from one cell to a full organism illustrates the power of cellular division and differentiation, which will be covered in few seconds. In the intricate hierarchy of biological organization, cells serve as the foundational units that differentiate into an astonishing array of specialized types, each with a unique role, structure, and function. Through a precise process of cellular differentiation, unspecialized cells called stem cells evolve into forms tailored to specific cells, each with specific structure, organelles, and function. These include osteogenic cells, fat cells, nerve cells, epithelial cells, muscle cells, and many others such as red blood cells. Later, cells of the same types come together to form tissues, each dedicated to a specific function. For example, osteogenic cells unite to create bone tissue, providing the sturdy framework that supports and protects our body. Fat cells merge into adipose tissue to store energy and insulate us against the cold. Nerve cells form nervous tissue, establishing a complex network that transmits vital signals throughout the body. Also, intestinal epithelial cells line the digestive tract, absorbing nutrients and shielding us from harmful substances. Moreover, muscle cells group to form muscle tissue, contracting to facilitate movement and generate force. After this, these different tissues then work in unison to create organs, each a finely tuned system. For instance, the heart pumps blood through coordinated muscle and nerve tissues. The intestines that blend epithelial and muscle tissues for digestion. The liver that filters toxins and synthesizes enzymes. And the brain that orchestrates our thoughts and actions through complex nerve. Finally, these organs integrate to form organ systems, such as the circulatory, respiratory, digestive, musculoskeletal, and nervous systems. This creates our body, ensuring it functions seamlessly. The plasma membrane is a crucial structure that surrounds the cytoplasm of the cell, acting as a protective barrier between the cell's internal environment and the outside world. The plasma membrane plays a vital role in maintaining the shape of the cell, transporting nutrients and waste, and facilitating cell signaling. With a few exceptions, cellular membranes, both plasma membranes and internal membranes, are composed of a phospholipid bilayer with embedded proteins, which provide the membrane with both flexibility and selective permeability. Before diving into the detailed structure of plasma membranes, let's first understand the precise structure of the phospholipid molecule. Phospholipids are composed of two fatty acid chains, a glycerol molecule, and phosphate group, attaching a polar group. As shown in this image, glycerol is a 3-carbon molecule that serves as the backbone of these membrane lipids. Within an individual phospholipid molecule, the fatty acids consist of one saturated and one unsaturated fat. As shown here, these fatty acids are attached to the first and second carbons of the glycerol group. Meanwhile, the phosphate group attaches to the third carbon of the glycerol backbone, and attaches various polar head groups. The phosphate group, glycerol, and polar group forms the polar head of the phospholipid, which loves water and said to be hydrophilic. On the other hand, the tail, which contains two fatty acids chains, is the non-polar part of the phospholipid molecules that hates water and described as hydrophobic. If we look at space-filling models of these molecules, we see their cylindrical shape, which allows phospholipids to align side by side to form broad sheets. In membranes, these phospholipids are often arranged in a double layer, as demonstrated in this image. In this arrangement, the polar or hydrophilic heads face the internal and external aqueous environments, which are the extracellular and intracellular environments, both of which are primarily water-based. On the other hand, the fatty acid tails meet together interiorly, which are hydrophobic, face inward, creating a hydrophobic core. Let's now discuss the precise structure of plasma membranes. The plasma membrane is made up of three major components. First are the lipids. Second are the proteins. And finally are the carbohydrates. These macromolecules work together to maintain the cell's structure and function. By this formation, the membrane is often described as a fluid mosaic. The term mosaic refers to the fact that the membrane is composed of a variety of different molecules, including lipids, proteins, and carbohydrates that are arranged in a patchwork-like pattern, creating a diverse and complex structure. It's called fluid because the components, such as lipids and proteins, can move sideways within the membrane, allowing it to remain flexible and dynamic. This fluidity is essential for the cell to adapt, communicate, and transport materials. Now let's move to talk about the structure and function of each membrane component. Let's start with the function of the membrane lipids. The phospholipid bilayer of the plasma first acts as a protective boundary, offering structural support and protection from harmful agents. It also helps shape the cell and functions. As a barrier, the phospholipid bilayer controls the selective permeability of substances entering and exiting the cell. Furthermore, the bilayer allows for flexibility and fluidity, which is important for cell signaling and communication. Cholesterol is another important component of the plasma membrane. It helps regulate membrane fluidity, stabilizing the membrane at high temperatures and preventing it from becoming too rigid at lower temperatures. Cholesterol also controls the permeability of the membrane, ensuring that the cell functions properly. The other component of membranes is the protein. Proteins make up roughly 50% of the mass of most cellular membranes, and they serve various essential functions. Membrane proteins are generally classified into two groups based on how they associate with the membrane. The first type of membrane proteins are the integral, or transmembrane proteins. As appears in this image, Integral proteins are embedded in the membrane and span across the lipid bilayer, and protrude from both sides of the membrane. If we examine the organization of transmembrane proteins, we see that they consist of three regions. Two hydrophilic regions, one on each side of the membrane, and a connecting hydrophobic region that spans the membrane. Importantly, transmembrane proteins cannot be separated from the membrane without disrupting the membrane itself. The other type of membrane proteins are peripheral proteins. These proteins are temporarily associated with the lipid bilayer or with integral membrane proteins through weak noncovalent interactions, such as hydrogen bonds. Notice the location of these proteins. You can recognize that peripheral proteins may be located on either the internal or external side of the membrane. In contrast to integral proteins, peripheral proteins can be easily separated from the membrane without damaging its structure. Let's now take a closer look at the functions of both the integral and peripheral proteins. Integral proteins serve a variety of functions in the membrane. Some of these proteins act as transporters, moving ions or other molecules across the membrane. Other transmembrane proteins serve as receptors, allowing the cell to receive signals from the external environment. Still others act as enzymes, catalyzing chemical reactions. Finally, integral proteins act as anchors, attaching to other proteins to help maintain the cell's structure and shape. Peripheral proteins play several key roles in the membrane as well. First, they can function as enzymes, catalyzing reactions at the membrane surface. In signal transduction, peripheral proteins often act as scaffolding proteins, organizing signaling complexes or interacting with receptor proteins to relay signals from the outside to the inside of the cell. Peripheral proteins also contribute to membrane support and shape by linking the membrane to the cytoskeleton. Additionally, they help regulate the movement of other membrane proteins, ensuring that proteins are properly positioned. Also, these proteins assist in the formation of lipid rafts, which are involved in cell signaling and protein interactions. The third major component of the plasma membrane is carbohydrates, which account for about 2 to 10% of the membrane's mass. These carbohydrates are primarily found on the outer surface of the membrane, where they form glycolipids and glycoproteins. Glycoproteins are proteins with attached carbohydrate chains, playing important roles in cell recognition, communication, and immune responses. Glycolipids are lipids with attached carbohydrate chains, contributing to cell signaling and forming the glycocalyx, a protective layer around the cell. Also, these carbohydrate-based molecules are crucial for maintaining the membrane structure, function, and ensuring proper cell interactions. At the end of this episode, let's understand why cell membranes are described as semipermeable. This is because it allows certain substances to pass through while blocking others. This selective permeability is essential for maintaining the cell's internal environment to preserve the cell. In this view, the phospholipid bilayer serves as a barrier that prevents the free passage of large, charged, or polar molecules such as amino acids, sugars, and ions. However, these large or charged molecules require specialized transport proteins, like channels and carriers, to regulate their movement in and out of the cell. In contrast, the plasma membrane is freely permeable to small, nonpolar, or uncharged molecules, like water, glycerol, and gases including oxygen, nitrogen, and carbon dioxide. As mentioned at the beginning of this episode, this selective permeability is vital for processes like nutrient uptake, waste removal, and maintaining a balanced internal environment.